Hello everyone, I hope you're having a nice day. It is evening here as I record, early November, cold and chilly. I hope your day is better. Let us look at, as we come to the end of our series on carbohydrate metabolism, let's look at glycogen metabolism. Did you know that our bodies store glycogen as a quick source of energy, mainly in liver and muscles? And when you're running, or exercising, your body taps into this glycogen as a continuous source of energy. So in this particular lecture, we want to know how the body is able to store this glycogen. How does it make it? And when or how does the body knows it is time to tap into this storage form? It is time to break glycogen and use it to store for energy, or it is time to make glycogen. Welcome to this lecture, and let's try and answer these questions. So a short reminder of glycogen. We know it is a polymer. It's a large branch polymer made up of glucose monomers. And these glucose monomers are joined by alpha 1 to 4 glycosidic bonds. And they also have multiple branch points made from alpha 1 to 6 glycosidic branch points. This structure of glycogen is very similar to the structure of starch in plants. However, one of the major differences is that glycogen is much more branched, approximately after every 8 to 12 residues of glucose held in alpha 1 to 4 glycosidic bond, we have a 1 to 6 glycosidic branch point. So this multiple branching allows the molecule of glycogen to appear spherical, and it also allows storage of large amounts of glucose monomers in a small space. And so many, and for example, we know the liver stores a lot of glycogen in the liver cells. Some interesting point about glycogen, it is predominantly found in the liver and skeletal muscle. The heart does make a bit of glycogen, but not so much. But the two major sources of glycogen are the liver and muscle. And one of the major differences is that the glycogen stored in the liver can be later on during periods of low blood sugar be broken down to free glucose and be released into the blood to be used by the rest of the body. However, the glycogen stored in muscle is only for muscle. And so it's only used for muscle needs. So maybe during exercise or intense exercise, that's when the glycogen is used to provide ATP. So let's start look at the processes that are there present in glycogen metabolism. So we have anabolic process of glycogen synthesis, and we have the catabolic process of glycogenolysis or breakdown of glycogen. Let us start looking at glycogen synthesis, which we refer to as glycogenesis. So as we say, this is how the glycogen molecule is made from individual glucose molecules, and it occurs in the cytoplasm of both liver and muscle. So there are three major steps. You can think about glycogen synthesis in three major steps involving activation of glucose, then elongation of the glycogen chain, and then formation of the branch points. And finally, we're going to see this process of glycogenesis is regulated by hormones, which are involved in blood sugar regulation of insulin, glucagon, and epinephrine. So the first step is activation of glucose. So why do we need to activate glucose? We know when glucose is activated, we increase its energy level and it becomes more reactive and can participate in reactions. So this glucose that is used in the synthesis of glycogen can be derived either from diet, what we have eaten, a high carbohydrate meal, or in gluconeogenesis. And we're going to see this later on when you discuss integration of metabolism, how the liver uses sugar from gluconeogenesis to restore the stores of glycogen in the period after fasting. We're going to see this. So glucose from either diet or from the process of gluconeogenesis can be used for glycogen synthesis. So the first thing we know when glucose enters the cell is that it is phosphorylated by the enzyme hexokinase to make it glucose 6-phosphate. So this glucose 6-phosphate, we have seen it can be used for glycolysis, but when the energy levels are enough and when adequate signals are there, glucose 6-phosphate is used for glycogen synthesis. How will it do this? The glucose 6-phosphate is isomerized to glucose 1-phosphate, where the phosphate group is moved from carbon number 6 to carbon number 1 of glucose to give us glucose 1-phosphate. The next step is to create UDP glucose, and here the glucose is attached to a nucleotide UTP, okay, by the enzyme UDP glucose phosphorylase, and the two phosphate groups from UTP are lost as inorganic phosphate, and we end up as UDP glucose. Sorry, one phosphate group from glucose and one from UTP are released as inorganic phosphate, and we end up with UDP glucose. So UDP glucose acts as the activated glucose, and it's the one who donates glucose for glycogen synthesis. So this is cool because even though we said glucose is the monomer that makes glycogen, it actually participates in glycogen synthesis in the form of UDP glucose. So once we have our UDP glucose, how does it now come together to form our glycogen? So before we start talking about how glycogen is elongated, we have what we refer to as a glycogen primer. In cells like the liver and the muscle, we have a protein called glycogenin, which serves as the glycogen primer. So glycogenin has an interesting structure. It is a protein, so it is transcribed and translated. It has two parts. It's made up of two different proteins held together, and it has an interesting activity which is referred to as autoglycosylation, meaning it is able to add glucose residues to itself. Okay, so as I had mentioned, it is a dimer. It has two copies of the enzyme, and it is thought, and there's evidence that shows these proteins glycosylate one another. So why is glycogenin important here? Is because it can autoglycosylate itself to up to 8 to 12 glucose residues, and it's a straight chain glucose residue. All glucose residues are held by alpha 1 to 4 glycosidic bond. So this is our primer that is needed for glycogen synthesis. So the next enzyme that is going to synthesize glycogen, it will be elongating the glycogen by adding to this primer molecule made up of the glycogenin protein with up to 12 H212 glucose residues. So this is very important to remember. So it's very interesting. You remember that picture at the beginning, we saw at the center there was a blue structure. It's a protein. So at the center of the glycogen molecule, we have the glycogenin protein. So glycogen chain elongation, as we have seen, it involves adding glucose residues already to an existing glycogen molecule. And the donor of this glucose, as we have seen, will be UDP glucose. But the enzyme that will catalyze this reaction is called glycogen synthase. So the glycogen synthase enzyme is going to add the glucose residue carried by UDP to the growing end of a already existing glycogen chain. So you can see this will be added here through an alpha 1 to 4 glycosidic linkage. And so the chain of glycogen will increase by 1 residue. Okay, so you can see here the split of the bond between UDP glucose will cause the release of free UDP. Okay, and we're going to create a new alpha 1 to 4 glycosidic bond, and the glycogen will grow by n plus 1 residue. So if it had 12, we'll have 12 plus 1, which is 13. So we have seen the glycogen synthase can only elongate by creating alpha 1 to 4 alpha 1 to 4 glycosidic bond. However, at the beginning we said glycogen is highly branched, and the branch points are at alpha 1 to 6 glycosidic bond. So this shows we have a second enzyme which is called branching enzyme, to summarize it, or amylo 1 to 4 1 to 6 glycosylase. So this enzyme is the one that is responsible for creating the alpha 1 to 6 glycosidic branch point. So what this enzyme does is that it is able to transfer a segment of about 6 to 7 glucose residues from a straight chain of alpha 1 to 4 linked glucose residues in glycogen and carry it aside to create an alpha 1 to 6 chain. So here you can see an example. Here was a short residue. Glycogen synthase added the glucose residues, okay? And as they became longer, the branching enzyme takes these residues and adds here a new branch, so that this chain now has become shorter, okay? And this one, a new branch has been created, okay? And it will continue growing the glycogen. So you can see now we have 3 points of elongation where glycogen synthase can now continue to create branch point. So we're saying that the segment to be transferred must be at least 11 residues away from another branch point. So this is very interesting. So the branches are not very close together, okay, to ensure proper spacing and structure. So you can see in glycogen synthesis, we have 2 major enzymes, the glycogen synthase and the branching enzymes that work together to create our branched glycogen with the alpha 1 to 4 glycosidic bonds and the alpha 1 to 6 branch points. Then we can talk about how this glycogen is broken down. The process of breaking down of glycogen is called glycogenolysis. Lysis, breakdown. Glycogenolysis. So here, glycogen is broken down back to glucose 1-phosphate. And of course, this occurs both in the liver and the skeletal muscle, but also in the cytoplasm, just the same site where synthesis occurs, in the cytoplasm. So this will be triggered when we are fasting or exercise, where there is increased requirement of ATP and a fuel to break down to provide that ATP, like in skeletal muscle. And it can be regulated also by insulin, glucagon, and epinephrine. So the enzyme that breaks down glycogen is called glycogen phosphorylase. So this enzyme can remove one glucose residue from glycogen and is able to release it as glucose 1-phosphate. Remember, the glucose residues in glycogen are not phosphorylated, but by the action of glycogen phosphorylase, we release one glucose residue that is phosphorylated on its first carbon, and that's why it is called glucose 1-phosphate. The interesting thing about glycogen phosphorylase, it can only break alpha 1 to 4 glycosidic bonds. Okay, so and we know glycogen has both alpha 1 to 4 and alpha 1 to 6. So it cannot break alpha 1 to 6, it only breaks alpha 1 to 4 glycosidic branch points. Another interesting thing about this enzyme is that it requires pyridoxal phosphate as a coenzyme. So this pyridoxal phosphate actually sits at the active site of glycogen phosphorylase and participate in the reaction of hydrolyzing a glucose residue from glycogen to create glucose 1-phosphate. And this glycogen phosphorylase is the main rate limiting step or the regulatory step, the enzyme that is regulated in the degradation of glycogen. So let's look a bit into the structure of glycogen phosphorylase. So the first thing is that the structure glycogen phosphorylase is a homodynamic dimeric enzyme, and it is made up of two identical subunits. In this diagram, we have this first subunit, which is green, and the other one, we've used two different colors to actually show the two subunits. Okay? Now, each subunit has an active site where it can be the site where glycogen is degraded. We also mentioned pyridoxal phosphate is actively involved in as a coenzyme for glycogen phosphorylase, and pyridoxal phosphate sits at the active site so that it can interact with the substrate. An interesting thing is that there is an N-acetyl glucosamine next to the active site that is thought to stabilize the pyridoxal phosphate as it sits within the active site. As this glycogen phosphorylase undergoes allosteric regulation, where we can have allosteric substances binding to it, it can transition between two states, the relaxed state and the tense state. The relaxed state is the active form, and this is the form that is actively breaking down glycogen. However, the tense form is the inactive form. There is a group of experimental drugs called chloroindole carboxamides. These are experimental, they are not yet in the market, but they have been found to bind at the dimer surface, and they are able to stabilize this tense state or tense conformation, which is inactive. And in experimental studies, they've been found to decrease glycogen breakdown, and they are used to regulate blood sugar, like for example in diabetics. However, they are not yet in use for humans. They are still at the experimental stage. So you can see if we have a drug that stabilizes the tense state, we can reduce the process of breakdown of glycogen. So similar to what we say with the glycogen synthase, the glycogen phosphorylase can only break alpha-1 to 4 glycosidic bonds. So we do require another enzyme to break the branching point of 1 to 6. So this enzyme is called the branching enzyme. So we say the glycogen phosphorylase can only cleave alpha-1 to 6 and alpha-1 to 4 linkages up to 4 residues from a branch point. You can see, for example, here. So here we have an alpha-1 to 6 branch point, and glycogen phosphorylase can cleave up to when you get 4 residues from this branch point. So this compound is called a limit branch. So the second enzyme, the branching enzyme, comes into place to remove the alpha-1 to 4. And this enzyme has two main activities. The first activity is that it is able to transfer these 4 branches, these 4 glucose residues, to the other side to create a longer chain with alpha-1 to 4 branches. So it is able to move a small oligosaccharide from a branch to a nearby linear chain, as we can see here. And so this is referred to as its transferase activity. The second activity is the glucosidase activity, with which it is able to cleave this branch point, the alpha-1 to 6 branch point. And the interesting part about this is that when it cleaves, unlike phosphorylase, it does not release glucose 1-phosphate, it releases free glucose. So the action of the debranching enzyme is that it cleaves and releases free glucose. So in summary, the combined action of glycogen phosphorylase and debranching enzyme will give us glucose 1-phosphate and free glucose. So the debranching enzyme, as we know now, it is the one that allows the breakdown of the alpha-1 to 6 glycosidic bond. And to summarize, we said it has two activities, a transferase activity and a glucosidase activity. So what happens to glucose 1-phosphate? We have another enzyme called phosphoglucomutase. So this enzyme does the isomerization of glucose 1-phosphate back to glucose 6-phosphate. We know glucose 6-phosphate can now used in metabolic pathways that generate energy. So this enzyme phosphoglucomutase catalyzation and isomerization of glucose 1-phosphate to glucose 6-phosphate. It does this thing in a very special way because at the enzyme has a serine residues at the active site which binds phosphate. So what happens through a two-step reaction, the enzyme donates the phosphate to the glucose 1-phosphate, and there's a time where it is both phosphorylated at carbon number 1 and carbon number 6. But eventually, the carbon number 1 gives the phosphate back to the enzyme, and we end up with glucose 6-phosphate. This is the end product. So even though it seems as a one-point reaction, there's that intermediate of glucose 1,6-bis-phosphate. So once glucose 6-phosphate ends up into the tissue, in muscle, it will definitely be used in the glycolytic pathway to provide energy because we have seen in muscle it is used when there's increased energy in muscle because of exercise. However, something interesting happens in the liver. How is the liver able to release glucose back into blood? It is because it has the enzyme glucose 6-phosphatase. This enzyme here, the phosphatase. We saw it in glucogenesis. So this phosphatase enzyme is able to release the inorganic phosphate from glucose to give us free glucose. And so this free glucose can be carried across GLUT2, across its concentration gradient, because like now we know the blood sugar is low and we're breaking down glycogen. That means as the glycogen is broken down, the hepatocyte or the liver cell will be having more sugar in their cytoplasm compared to the amount of sugar in the blood. So if glucose 6-phosphate is dephosphorylated back to glucose, glucose will use the same GLUT2 receptor that it had entered the cell with to come out of the cell into the bloodstream from a region of high concentration to a region of low concentration. This enzyme is lacking in skeletal muscle, and so muscle cannot release its bloodstream, its sugar back into the blood. So how is glycogen metabolism regulated? So the interesting thing we have seen, both of these pathways occur in the cytoplasm. And if they are not regulated, besides occurring in the cytoplasm, they occur spontaneously and they are thermodynamically favorable. And so if we had one corner of a hepatocyte synthesizing glycogen and another corner breaking it, it will result to what we refer to as a futile cycle. So to prevent this futile cycle, glycogen and synthase and glycogen phosphorylase are reciprocally regulated. We have seen this word again, reciprocal regulation, when we talked about phosphofructokinase 1 and fructose 1,6-bisphosphate regulation in regulation of glycolysis and gluconeogenesis. The two of them were the key rate limiting steps in the other pathways. So we had to make sure glycolysis is not occurring at the same time as gluconeogenesis. Something similar happens here, and this is mainly through allosteric effectors and phosphorylation. Let us look at these mechanisms. So how does allosteric regulation influence the activity of glycogen phosphorylase? We can start at looking at this in the muscle. So at AMP, which is an indicator of low energy levels, activates glycogen phosphorylase in muscle. So when glycogen phosphorylase is bound by AMP, it shifts to its relaxed state, and this relaxed state, we said it is the active state, and it can break down the stored glycogen. However, when we have high energy levels, as indicated by high levels of ATP or high levels of glucose 6-phosphate, these ones will inhibit the phosphorylase enzyme. Okay? They will inhibit it by stabilizing the tense inactive form. So when they are bind to the allosteric site, they will convert glycogen to the inactive tense form, and so this will limit the breakdown of glycogen. What happens in liver? So in liver, there is an phosphorylase isoenzyme, which is slightly sensitive from the muscle isoenzyme, in that it is less sensitive to the regulators like AMP, ATP, and glucose 6-phosphate. Remember, the liver is not doing a lot of contractility actions like skeletal muscle. However, in liver, glycogen breakdown is inhibited when ATP and glucose 6-phosphate are high. So even though it is less sensitive, doesn't mean it is inactive, but it is still active, but it's not as sensitive to the levels of these modulators that are very active in skeletal muscle. Then how is glycogen synthase regulated? It also undergoes allosteric regulation. And now this one is activated by glucose 6-phosphate, showing we have enough substrate. So glucose 6-phosphate we have seen is the one of the substrate. It is converted to glucose 1-phosphate, then it is used to form glycogen. So when we have high levels of glucose 6-phosphate, they indicate high blood sugar, and so this enhances glycogen synthesis. Okay, so also we'll have glycogen synthesis when ATP levels are high. So remember, when glucose enters into the cell, the predominant reaction is to make energy in the form of ATP. But like in the liver cell and skeletal muscle, if you have ATP in adequate amounts, so there's a sufficient energy, so the extra glucose 6-phosphate will be used to make glycogen, which can be used for later, can be reserved for later use. Another way we've mentioned that of regulation of the activity of glycogen synthase and glycogen phosphorylase is through phosphorylation. And this means we can phosphorylate the enzyme and we change its activity or dephosphorylate. So this kind of mechanism of regulating enzyme activity, we call it covalent modification. And hormones play a very big role in this. So if we start looking, hormones glucagon and epinephrine are stimulated when the blood sugar is low. So for example, glucagon, the blood sugar is low, glucagon is produced to try and stimulate processes that will bring up the blood sugar. Epinephrine could be triggered during fight or flight responses. You're scared, there's a lion. Its role is to make sure there's adequate sugar in the blood for any activity you will take, whether you want to run away or you want to fight. Epinephrine, it does that. So either way, they will stimulate. One of the ways they'll do that is to stimulate the breakdown of glycogen to bring the blood sugar in the blood high enough. So glucagon predominantly affects liver breakdown of glucose, whilst epinephrine can act in skeletal muscle. One of the common things about them is that they activate increased cellular concentration of cyclic AMP. So how does this do? So when the hormones, either epinephrine or glucagon, binds to the receptors through activate the formation of cyclic AMP, how do they do this? So through their receptors, which are G protein coupled, they're able to activate the enzyme adenylate cyclase, which is inactive, to adenylate cyclase, the active form. So this adenylate cyclase is the one that is able to make cyclic AMP. So the ATP will be split to cyclic AMP. And so the signal we are going to see is an increase in intracellular cyclic AMP in both the liver and the muscle. Increase in CAMP will cause activation of several enzymes, and one of the enzymes that is activated is protein kinase, which when lower CAMP concentration is inactive, but once the intracellular CAMP concentration increases, it becomes activated. So we can say the name kinase, so it must be an enzyme that phosphorylates. So protein kinase A phosphorylates other enzymes, and one of the enzymes it's going to phosphorylate is phosphorylase kinase. Phosphorylase kinase is a specific enzyme that phosphorylates glycogen phosphorylase. So the phosphorylase kinase will be activated by protein kinase from converting it from inactive form to the active form. And how is this done? By phosphorylating it. So protein kinase will phosphorylate phosphorylase kinase and make it active. Now, once it is active, it can now go and phosphorylate glycogen phosphorylase from its inactive form to its active phosphorylated form. So the active form of glycogen phosphorylase is the A form, which is phosphorylated. So you can see it looks like a long cycle, but remember, every time in this cycle, one molecule of epinephrine will bind to its receptor. It will activate several adenylate cyclase. This will activate several cyclic AMP. This will phosphorylate will activate several protein kinase A, and this one will phosphorylate several phosphorylase kinases. So the effect of this is that it amplifies the effect of the hormone, so that by the end of the day, once the hormone epinephrine or glucagon is produced, you have several molecules of phosphorylase, glycogen phosphorylase activated, and they actively break down glycogen, and they will make sure there's blood sugar is high or there is glycogen adequate for muscle contractility. Then how is glycogen synthase activated? So for glycogen synthase, remember it's the one that makes glycogen. So when we have glucagon and epinephrine, we have seen it is going to activate protein kinase A. And protein kinase A will phosphorylate several proteins. And one of the proteins that will be phosphorylated by protein kinase A is glycogen synthase. So we know once glycogen synthase is phosphorylated, it moves from its active form to its inactive form, the B form. So it is less active. So once we have glucagon and epinephrine, as they activate glycogen breakdown, they will inhibit glycogen synthesis by phosphorylating glycogen synthase, and this inhibits glycogen synthesis, for converting it to the B form of the tense form, which is less active. However, high levels of glucose 6-phosphate, especially in the liver, can inhibit this action of glucagon and will activate glycogen synthase and will promote glycogen synthesis. Now, how does the glycogen synthesis start? So we have an enzyme called protein phosphatase, which is able to remove the phosphate group from glycogen and convert it to its A form, which is the active form. And this protein phosphatase can be activated by high levels of glucose 6-phosphate, which is the substrate for glycogen synthesis. So how does insulin work? So when we have high blood sugar, insulin is produced, and via several mechanisms, it's going to activate phosphoprotein phosphatase. So this enzyme will go around the cytoplasm dephosphorylating proteins. And one of the enzymes that is dephosphorylated is glycogen synthase, which activates it. And glycogen phosphorylase, when it is dephosphorylated, it becomes inactive. So in this way, this enzyme phosphoprotein phosphatase counteract the effect of the CMP cascade, but also the combined actions of insulin allows regulation of these two pathways, such that when synthase is active, phosphorylase is inactive. In skeletal muscle, in addition to the hormones, epinephrine, we also have calcium playing a very integral role in regulating glycogen breakdown. So we know during contraction, calcium is released from the sarcoplasmic reticulum, and this is mainly, we know, it facilitates a binding of actin to myosin. But also, calcium is taken up as a signal to indicate increased requirement for energy. And one of the things that calcium binds to, it binds to a protein called calmodulin. Calmodulin in skeletal muscle is a subunit of phosphorylase kinase. So that means phosphorylase kinase has calmodulin as its subunit. And so when calcium levels bind to calmodulin, it activates this protein, phosphorylase kinase. Okay? And so phosphorylase kinase now phosphorylates glycogen phosphorylase, which will stimulate degradation of glycogen. So during extended exercise, as you have said, glycogen stores are depleted, and if you exercise for a long period of time and the muscle has to look for other sources of energies from the blood, like maybe glucose that can be formed from glucogenesis, or fatty acid in aerobic metabolism. So remember, this calmodulin is a part of the phosphorylase kinase, and when the intracellular calcium concentration increases as during contractility, okay, binds to calmodulin, which activates the phosphorylase kinase, And now the phosphorylase kinase can go and phosphorylate glycogen synthase, glycogen phosphorylase, which will break down glycogen to glucose, which can be used for ATP synthesis. So there are several glycogen storage diseases which results from defects in the enzymes that are involved in the synthesis of glycogen. So the results of this could be abnormal storage of glycogen in either liver, muscle, or heart because of increased accumulation of glycogen. So the symptoms of these glycogen storage diseases, depends on which isoenzyme is affected. Is it the one in muscle or is it the one in liver? So when it is a problem with the muscle glycogen pathway, most features will be muscle weakness. But if it is the liver, because we know the liver glycogen is released into blood, we are likely to have symptoms like hypoglycemia. If you're storing large amounts of glycogen in liver, we are likely to have hepatomegaly and overall liver dysfunction. So here is a small table highlighting some of the glycogen storage diseases. A common one is Von-Gehr's disease, where we have deficiency of the phosphatase enzyme, the glucose-6-phosphatase, especially in the liver and kidney. Remember, this enzyme is only found in gluconeogenic tissues. And so if it is absent, we're not able to release glucose back to the blood, especially during periods of fasting. And so one of the features that are going to be present is hypoglycemia, especially when someone has not eaten. But also because glycogen accumulates in the liver, we end up having hepatomegaly. And because the body looks for other sources of fuel, so it will end up doing aerobic glycolysis, so we'll have increased lactic acid levels and increased catabolism of lipids, causing ketosis. And the glucose-6-phosphate, which cannot be released, might enter into other pathways like pentose phosphate pathway that will form purine nucleotides, which when they are broken down, they form uric acid and contribute to hyperuricemia. We have Pompe's disease, where we lack a lysosomal enzyme that breaks down glycogen in lysosomes, called alpha-1 to 4-glucosidase or acid maltase. And that means glycogen tends to accumulate in tissues, and it can cause cardiac failure, especially in infants. So I would request you go through these diseases, Corey disease, Anderson's disease, and look at the symptoms and try to determine which enzyme is missing and whether it is present in muscle, and identify the differences in the different enzymes, different symptoms, and relate them to the point of deficiency of the enzyme. Otherwise, I would like to stop here. Thank you so much for your time. Have a lovely time. Have a good one.